Hello everyone. So from in this particular class, we are starting cell biology chapter. See friends, whether you are preparing for the JL or DL, whether it's a botany or zoology, we know very well the cell biology is the most common chapter. From the cell biology, we have to focus on cell organelles, cell division, then followed by cell cycle regulation. These are all the three subtopics we have to focus on the chapter of in the chapter of cell biology. So this chapter is a very high weightage chapter for both botany as well as the zoology. So friends, in this particular chapter, I will solve 200 to 250 questions from only cell organelles, from very basic level to the advanced level of questions. Similarly, friends, we will solve 200 to 250 questions from the cell division and cell cycle regulation, from very basic to the very advanced level. So this particular cell biology, I will continue simultaneously along with the plant physiology. It will take 5 to 6 classes to complete this particular chapter because it's a very big chapter. So in this particular class, we will study basic level as well as the moderate level of the questions. Okay, now let's start. So the question number one, the main area of cellular activities in the plant and animal cell. I have explained it to all of you many times. Cytoplasm is the region. What happens in the cytoplasm, friends? Most of the biochemical reactions will be taking place in the cytoplasm only. Many biochemical reactions. Let's take an example, protein synthesis. Let's take an example, the most important example, the glycolysis. So many key reactions are happening in the cytoplasm only. So friends, the answer will be the cytoplasm. This is the main area which is involved in the cellular activities. Second question, the cell is considered to be living in the living state when it possesses. This is also a very interesting question. What most of the students will think, if nucleus or the DNA is present, they will think that then the cell is said to be the most important one. See friends, every one of you listen. Suppose this is the cytoplasm, which is a fluid like structure. In the cytoplasm only all the organelles are situated, all the organelles are embedded. So you have to understand the point. Without the cytoplasm, there is a no nucleus can be situated. Within the nucleus only DNA can be situated. If you take a prokaryotic organism also, the DNA is present in the cytoplasm only. So always remember, the cell is considered to be in living state when it possesses cytoplasm, not to get confused with that. Then the question number three, the cell organelles commonly present in the prokaryotes also and eukaryotes also. So the very common answer will be the ribosome, because you know very well the lysosomes are not present in the prokaryotes. They contain some enzymes. Nucleolus is not present in the prokaryotes. Ribosomes will be present in the prokaryotes also as well as the eukaryotes also. It contains 70s ribosomes. Eukaryotes contain 80s ribosomes. But the ribosomes are commonly present in the both prokaryotes as well as the eukaryotic organisms also. Question number four, very simple question. Non-membranous bound organelle found in both prokaryote and eukaryote. You know, right? Nucleus having the membrane. First of all, anyway, plasmid is also surrounded by a, membranous structure only, but it's a single layer. Chromosomes, you should not call chromosome is a membranous and non-membranous. No, you should not call that. So you can eliminate the option of chromosome. How can you define the chromosomes with respect to the structure, friends? You have to understand condensed and relaxed forms. You should not say that membranous and non-membranous. So the question is non-membranous bound organelle present in both. So undoubtedly, friends, ribosomes, which plays a crucial role in the protein synthesis, which is not covered by any kind of a membrane. The cell organelle in the animal cell that helps in the cell division. We all people know that centrioles or the centrosomes, which are helpful in the cell division. Is it? It's a direct question. And remember, everyone try to understand the question number 6. Choose the correct answer. Very good question. All the prokaryotics have cell wall surrounding the cell membrane except in mycoplasma. We know, right? Mycoplasma do not have any kind of a cell wall. That's why we can call it jumping genes of the microbiology. not to get confused. So option 1 is absolutely true option. Option 2 says that all prokaryotes possess a double membrane cell organelle. That is wrong, right? Prokaryotes do not contain chloroplast. Prokaryotes do not contain mitochondria. Prokaryotes do not contain nucleus. So it do not contain double membrane cell organelles. Option 3, prokaryotes are identified by membrane bound nucleus. No, not at all. It is do not contain the nucleus only. Option 2 is wrong. Option 3 is also wrong. And histone proteins are seen in the prokaryotes. That is also absolutely wrong statement. So which is the only correct statement? Option 1. All prokaryotes have cell wall surrounding cell membrane except in mycoplasma. Easy to remember. Question number 7. The component of the bacterial flagella are. Very simple question and very direct question. Filament, hook, and basal body. These three are the three common components of the bacterial flagella. Every student commonly know that. What's the ultimate function of the flagella in the bacterial body? Of course, friends, it plays a key role in the motility. We all people know it. And we also know that some bacteria having monoflagellates, some bacteria having diflagella, some bacteria throughout the body, they contain the flagella only. But the common components are filament, hook, and basal body. Direct question, straightforward question. In the yesterday class only, I have explained you the question number 8. The bacteria on its outer membrane, it contains glycocalyx. What did I told you, friends? If the glycocalyx is attached very, very tightly, we call it as the capsule. If the arrangement is the loose arrangement, you call it as the S layer. You can also call it as the slime layer. Obviously true. So tight or condensed structure is the capsule. But in the question they have asked loose structure, which is the slime layer. Not to get confused with that. A direct question, friends. Even this question asked in the set level also. Triple helical structure of the collagen was discovered by. So Ramachandran. You know, right? Ramachandran is the one who proposed the Ramachandran plot for all the amino acids. Right? So anyway, triple helical structure of collagen also discovered by Ramachandran. Next one, question number 10. The cell membrane is said to be dashed with respect to the movement of molecule across it. We all people know very well whether mitochondria contain cell membrane, chloroplast contains cell membrane, nucleus contain the nuclear membrane. All the membrane specialties what? They are not allowing every single ion or every single molecule across the plasma membrane. They are moving only a selective ions across the plasma membrane. So what will be the answer? Selectively permeable. Question number answer is 4. Question number 11. The approximate percentage of protein and lipids in the membrane of RBC cell. Friends, in the yesterday class, I have explained to all the students. What I have explained to you, the composition of the percentage of protein and lipid is not the same for every cell. That's a very important statement. It will be varied from cell to the cell, not to get confused with that particular point. So since they have asked the question with respect to what, friends? RBC. So in case of RBC, the percentage of protein will be 52% and lipids will be 40%. Not to get confused with that. Next, one of the following is an important constituent of the cell membrane. They did not ask the cell wall. Cellulose, calcium pectate, lignin, these three belongs to the cell membrane. And what we discussed, friends, cell membrane is majorly consist of the phospholipids. Hence, the correct answer is 3. Question number 13 says that passive transport of molecule across the membrane implies that movement occurs. Not to get confused. Every student commonly know that. Passive transport means what, friends? The movement of ions from high concentration to the low concentration. Are we spending any kind of energy? Without energy requirement. Direct straightforward question. So what will be the answer? Undoubtedly, without energy requirement. Option 2 is wrong. Irrespective of energy requirement, that is also wrong. From low concentration to high concentration, it is always move from high to low. Hence, the correct answer is what, friends? Option 3 is the most appropriate answer. Question number 14. The components of the cell membrane that cannot be attributed to its fluid nature. Every student remember the question very correctly. The very simple question, friends, but very logical question. the components of the cell membrane that cannot be attributed to its fluid nature. In the yesterday class also, I have explained it to every one of you. The plasma membrane plays a crucial role in the fluidity. How? With the help of lipid molecules, with the help of proteins, as well as with the help of cholesterol also. What is the question they are asking? Among these, which is not involved in the fluidity? Peripheral proteins involved in the fluidity, carbohydrates involved in the fluidity, and which is not involved in the fluidity? Intrinsic protein, which we also call it as what proteins? Integral proteins. They are not involved in the fluidity. Hence, what is the correct answer? Answer is 1. Simple question, but very important question. One of the following statement with regard to the movement of the neutral solute across the membrane is incorrect. Friends, they are asking which statement is incorrect. Right. It takes a passive transport. No, it is a correct statement. It takes place by simple diffusion. That is also true statement. It occurs along the concentration gradient. That is also true statement. Friends, neutral solute, how they will follow? They will follow by the passive diffusion, which means what? No requirement of energy. How they are moving, friends? They are moving from high concentration to low concentration. That is only we can call the simple diffusion or the direct diffusion. It do not require any energy. So option 4 is the incorrect option. Simple question. Question number 16. A carrier protein, we will discuss in the transport of the plasma membrane. A carrier protein is required for the transport of these ions across the membrane. Very simple question, right? Means carrier protein is required for the transport of which molecules? Undoubtedly polar molecule. Because friends, neutral molecules will show the simple diffusion. Non-polar molecules will use the active transport. I'm sorry. Non-polar molecule also will show the direct diffusion because they can easily cross the membrane. Which cannot cross the membrane? Polar molecules cannot cross the membrane. So they are using what? A particular carrier. That carrier is involved in the transport of that molecule. But are we using energy? No. It is called as what mechanism? We will discuss in the plasma membrane transport. Facilitated diffusion. We are not using any energy. But the movement of ion or molecule is happening just with the help of carrier protein. Hence the correct answer is 1. Transport of sodium, potassium across the membrane. It's a direct question. Many number of times you studied, it is an active transport. Correct, na? If 3 sodiums are coming inside, 2 potassiums will go out of the cell. And it also varied from cell to cell only. But sodium, potassium, how they will work, friends? They will work with the help of ATPase pumps. So friends, undoubtedly ATPase pumps are dependent on the energy, which is an active transport. Not to get confused, the non-living layer forming the covering of the fungal cell is obviously what it is? Cell wall. See, non-living layer forming the cell covering of fungal cell. You know, right? What does the fungal cell wall is made up of? It is made up of the chitin. Correct, na? What is chitin? Which is a polysaccharide, which is made up of a NAG and a NAM sugars. Right, which is a non-living layer, which is the chitin and which is the cell wall. It's not cell membrane. Tonoplast is present where? It is present on the vacuole. Capsid is completely irrelevant option. Now, question number 19, think and answer. One of the following statement with respect to the primary cell wall is wrong. They are asking wrong. It is seen in the young plant of cell. Absolutely true. No doubt about it. Actually, friends, remember my words correctly. Once the cell become matured, what happened? Primary cell wall only converted into the secondary cell wall once it reaches to the maturation point. Is it or not? So option 2 says that it is first formed between middle lamella and secondary lamella wall. That is wrong statement. Just now I am telling to all of you, right? First primary cell wall will formed before the secondary wall. But option 2 says that it is formed between the middle lamella and secondary wall. That is wrong statement. It is capable of growth. Yes, of course. It is present between the middle lamellae and the secondary cell. See, not to get confused with this particular option 2 and option 4. Most of the students are getting confused here. Here they just said secondary cell, not the secondary wall. So primary cell wall is present between the middle lamella and secondary cell. Later, at the maturation stage, the primary cell wall divided into secondary cell wall, or sorry, differentiated into secondary cell wall, not to get confused. Option 4 is also right option. Option 2 is the wrong option. Question number 20. The layer of the cell wall that develop from the cell plate during the cell division. We all people commonly know that. Primary cell is present in the middle only. Later it is differentiated into secondary cell and it is not at all the tertiary cell. So they're asking which cell wall, the layer of the cell wall which formed from the cell plate. Undoubtedly, friends, which will be the middle lamella. I want you to remember, when I am explaining you the cell wall, I will explain you briefly. Next one, question number 21. The cytoplasmic bridge that connect the cytoplasm of two neighboring cell. This is one cell, this is another cell. The cytoplasm of the two cells can be connected with each other by a cytoplasmic bridge. What do we call that particular cytoplasmic bridge, friends? We can call plasmodesmata. In fluid mosaic model of the plasma membrane, we studied in the last class. What we discussed? What is fluid mosaic model? A variety or a collage of proteins are embedded on the lipid. Upper layer is non-polar and hydrophilic. Of course, true, right? This is the lipid molecule. What is this? Non-polar. I'm sorry. The question says that in fluid mosaic model of plasma membrane, this is wrong statement, right? It is a polar. I'm so sorry, friends. The statement is wrong statement. Here, upper layer is the polar. Here, upper layer talking about the head and tail of the lipid. I'm so sorry. See, this is the head of the lipid and this is the tail of the lipid. And what is the head? Head is the polar in nature and hydrophilic. What about the tail? Tail is the non-polar. And what it is, friends? It is the hydrophobic in nature. Correct, na? I have written wrong here. This is the correct statement. Polar and hydrophilic. Option 2, polar layer is hydrophobic. No, right? It is the hydrophilic. Phospholipids form a bimolecular layer in the middle part. We can't say that middle part. What we can say that phospholipids will form a bimembranous layer, not in the middle part. Because of the phospholipid only, the membrane will form. It is not forming in the middle part. Proteins forms a middle layer. Proteins never form layer, first of all. Proteins always integrated, either in the inside, which we can call integral protein, or in the peripheral, on the outer or in the deeper inner. They never form a layer, first of all. Hence, the correct answer is fluid mosaic model. Upper layer is a polar and hydrophilic in nature. That is the true statement. 23. Plasmodesmata connection helps in. What is the use of plasmodesmata? We discussed, right? It is connecting the cytoplasm of two cells. What is the use? Involved in the movement of ions or molecule from one cell to the another cell. Obviously, option 4. As I already told you, friends, it will be differ. The ratio of the protein and the lipid content will be differ from cell to the cell. So here they are asking about the ratio, not the percentage. In previous question, we have seen is the percentage. Now here we are seeing is the ratio. Here I am requesting you to remember the ratio. So which is 10 is to 13. It's a direct question. You have to remember that compulsory. Percentages and ratios we have to remember, friends. Question number 25. Continuity of cytoplasm from cell to cell is maintained through the cytoplasmic connection. I think question got repeated, which is plasmodesmata. Tight junction is what, friends? It will seal the gap. Gap junctions are also involved in the movement, but gap junctions are not connecting the cytoplasm of two cells. Cytoplasm of the two cells can be connected only by the plasmodesmata. Question number 26. The main difference between the active and passive transport across the cell membrane is that I told you very clearly. Passive transport means along the concentration gradient, high to low. Active transport means low to high. Passive transport, we don't use energy. Active transport, we use the energy compulsory. So passive transport is active, wrong statement. Active transport is passive, wrong statement. Passive transport is gradient based, while active transport is energy based. Of course, true. Option 3 is the right option. We can say that. Option 4, passive transport is confined to anions. No, it's not like that. Passive transport can move involved in the movement of any kind of ion or any kind of a molecule without using energy along the concentration gradient. Question number 27. Neutral solutes once again may cross the membrane. Neutral solute can cross the membrane directly. Simple diffusion. Simple question. Along the concentration gradient, not against the concentration gradient. They are all very basic questions. The moment in the next classes, we will see the advanced level and tough questions. Which element mainly occurs in the middle lamellae, which is the calcium? Of course, calcium pectate. Cell wall is lignified. Cell wall of lignified cell is lignin. It's a kind of a dead component. It's a dead component. And friends, you know, right? Cell wall is permeable. Which is selectively permeable? Selectively permeable is the plasma membrane. So it is not semi-permeable. It is not impermeable. Not impermeable. Which is the permeable and dead. Very direct question. Answer is 2. Question number 30. The cells are held together by a calcium pectate layer is called as. Of course, where the calcium pectate is present, you studied in the 10th standard also, present in the middle lamella. Ingestion of the solid food by plasma membrane is called as. What do we call it, this particular process, friends? We can call phagocytosis. I will explain you the type of phagocytosis when we are reading the plasma membrane transport mechanism. There we will explain the types of phagocytosis. I mean to say, what do we can call liquid digestion? What do we can call the solid digestion? We will discuss that all elaboratively. Ingestion of a large molecules by animal cell is called as. They're asking, friends, animal cell, not the plant cell. They're asking ingestion of larger molecules. So ingestion means what? We are taking, right? Which is a kind of endogenous process. Correct, na? Not to get confused with this particular question, listen to me very correctly. Ingestion of the large molecules by animal cell. Remember my words. Suppose example, this is the receptor. Example, a food particle or the pathogen got integrated, right? When the pathogen got integrated, listen my words very correctly. So you take a best example of macrophage. This is the macrophage. This is the pathogen. So the pathogen trapped by the macrophage, and what process it will undergo? It will undergo the receptor mediated endocytosis, and it is packed in the vesicle called as a phagosome. Correct, na? So what do we call this particular process? We can call it endocytosis. We are taking it inside. So which is called endocytosis. And friends, not to get confused with the question number 31, you can call pinocytosis also, you can call phagocytosis also. But most appropriate answer will be the phagocytosis only. If they did not given the phagocytosis, then you can go for the pinocytosis. Not to get confused with that point. So pectinage helps in ripening of a fruit. It dissolves what? See, the question is a very simple question and logical question. They are given a question that pectinase helps in the ripening of fruit. Friends, you all people know that. Why the fruit get ripened? You tell me. Fruit become ripened when the cell wall become degraded, when the cell wall become loosened, when the cell wall become completely destructed or degraded. So pectinase help in the ripening of fruit by dissolving what? Undoubtedly calcium pectate, which is present in the middle lamella. Once calcium pectate got degraded, the cell middle lamella will become very loosened. Then obviously the fruit starts becoming ripening very faster. Not to get confused with that. Okay. Okay. Question number 34. Select the structure to synthesize more amount of the cellulose. This is a very good question. Most of the students won't understand this particular question. The question says that which synthesize more amount of cellulose. Plasma membrane never synthesize. You know very well endoplasmic reticulum is involved in the synthesis of protein. SCR involved in the synthesis of lipid. See, I can't say that highest amount, friends, a minimal amount. The Golgi complex involved in the synthesis of the cellulose by an enzyme called as cellulose synthase enzyme. Right, where one glucose can be added with another glucose, like that 20,000 to 40,000 of glucoses will add and give rise to the synthesis of cellulose. Answer is number 1. 35. Which one of the following act as a direct question, right? Cementing material. Undoubtedly, without thinking, calcium pectate. Sodium is ion, wrong statement. Lignin function is what? It is involved in the protection and mechanical strength. Cementing material is calcium pectate. 36. Bacterial cell wall is composed of. It's also a very good question. Cell wall is made up of protein also. Cell wall is made up of the lipid also. Non-cellulosic carbohydrate is a wrong statement. Hence, the correct answer is number 4. Which of the following process? See, friends, this is also very simple, very logical question. Which of the following processes include all others? Let's take passive transport. What is passive transport? Tell me. High concentration to the low concentration without spending any energy across the membrane. So, under the passive transport, osmosis will come. Movement of water molecules, diffusion of solute, simple diffusion, facilitated diffusion. In any of these three, we don't use any energy and always moving along with the concentration gradient only, right? Hence, the correct answer is 4. 38. Which of the following statement is not, they're asking not true. It is present in both plant and animal, right statement. Lipid is present as a bilayer. Always remember, lipids never present in the middle, friends. Proteins are present integrated as well as loosely associated with the lipid bilayer. Yes or no? Which will loosely associate? Peripheral. Which will be integrated? Integral protein. We already know it. Option 3 is also right option. Option 4. Carbohydrate is never found. That is wrong statement, right? 8 to 10% of the plasma membrane generally contain carbohydrates. So option 4 is the wrong option. 39. The position of the middle lamellum in a typical plant cell is. Friends, they're asking location of the middle lamella, where the middle lamella is present in between two primary cell walls of adjacent cells. Undoubtedly true. Right statement, no need to think at all. It never located inside the primary cell wall, inside the secondary wall, and it never located outside the membrane. It is always located in between two primary cell walls of two adjacent cells. This is the true answer. And we all people know that apoplast is, you know, right? There are two type of transports are there. Symplastic transport, apoplastic transport. Symplastic transport means every student commonly know that via plasmodesmata. Apoplastic means through the cell wall and intercellular space, which is a straightforward and direct question. So what will be the answer? Undoubtedly, intercellular spaces containing non-living matter, because it is not connected to any adjacent cell. Obviously, 4 is the straightforward answer. 41. The model which describes the plasma membrane as a protein iceberg in a sea of lipid. Obviously, which model? Fluid mosaic model. Yes or no? We studied very correctly. Lipids are acting like a sea, and on the sea, proteins can be associated like a icebergs. So, which is the number 1? Desmosomes, clear cut question, involved in the process of. All the students listen. Desmosomes and hemidesmosomes, both are, what do we call them as? We call them as junctional molecule. Junctional molecule function is what? Cell to cell adherence or the cell to cell attachment. Hence the answer is 3. Easy to remember. Question number 43. Cell organelles in the endomembrane system are coordinated with each other. That's a very good question, right? Cell organelles in the endomembrane system are coordinated with each other. Of course, functionally, outer membrane, RER protein, SER lipid. So they are coordinated with each other with respect to what? Function, not by structure. Some students are getting confused with that. Not by structure, it is by function. Number 1. Which of these cell organelles is not considered as endomembrane system? Some students will answer ribosomes. No. They are asking, see, this is RER. RER connected via SER. I mean to say both are interconnected with each other. I am saying on the top of RER, ribosomes are present. So it is not the part. Never get confused. It is not the part. And you know, right? Whatever the proteins that are synthesized, finally they move to the lysosome only with the help of Golgi complex. So friends, ER, Golgi complex, RER, SER, Golgi complex, lysosomes are associated, but not the ribosome. Ribosomes are the integral parts. They are not asking integral part, which is not embedded. Sorry, which is not the part of endomembrane system, which is the ribosome. You know, right? Ribosomes do not have any membranous structures also. Question number 45, direct question. The major site of the lipid synthesis is SCR. Not only lipid, you can call cholesterol, you can also call sex hormones. All can be synthesized on the smooth endoplasmic reticulum. One among the following is considered as the part of the endomembrane system. Peroxisome, chloroplast, mitochondria, they are not at all the part of the endomembrane system. Which is the part of endomembrane system? Which is undoubtedly, friends, vacuole. The endoplasmic reticulum that is not studded with ribosome on the surface of the surface is the major site for the synthesis of, which is SCR. SCR involved in the synthesis of lipids. They are basic, but we have to know it. Then only you can understand the advanced level questions. 48, the cell brain shows extensive and continuous tiny tubular structures on its outer membrane is called as. What is that? Undoubtedly endoplasmic reticulum. What is the cell brain here? Nucleus, right? Every student know this is the outer membrane of nucleus. We will study, and this is the inner membrane of nucleus. Outer membrane of nucleus only connected with the rough endoplasmic reticulum, and that rough endoplasmic reticulum only contains the ribosomes. So answer will be RER, that is the endoplasmic reticulum. And remember, smooth endoplasmic reticulum never connected to the nucleus. That is also a very important point. Only RER will connected to the nucleus. 49, the cell organelle that is actively involved in the protein synthesis is continuous with. In this question only I have explained it, right? It is always connected to the outer membrane, not to the inner membrane, not to the inter. Always to the outer membrane of nucleus, not with any other organelle. Packing of materials is the principal function of packaging. Sorting is the principal function of Golgi complex. Endoplasmic reticulum involved in the synthesis. Lysosome contain degradative enzymes. Vacuole can act as a storage organelle. Is it or not? These are all very basic level questions, friends. In the tomorrow class, we will study the advanced level of the questions. Thank you. Thank you all.